Alright, we will look at mutation now. So you look at gene mutation, right? There are three types: addition, deletion, which may result in frame shift, and substitution, which may result in silent, missense, or nonsense mutation. Alright, so the mechanism of the change in the gene is through either addition, deletion, and substitution. The result of the change is a frame shift, or silent, missense, and nonsense. Okay, so gene mutation is defined as a change in the DNA sequence of the gene, right? So you can imagine, right, if the DNA sequence of the gene is changed, then the sequence of the template strand is changed. So because of that, after transcription, the mRNA sequence will change. A change in the mRNA sequence may result in a change in the amino acid sequence after translation, right, based on the mRNA that is changed, right? So there may be a change in the amino acid sequence, which is a change in the primary structure of the protein. If the primary structure of the protein is changed, and remember that primary structure determines the shape of the protein, right? So there may be a change in the 3D conformation of the protein, which may change the protein function. And if the protein functions differently, then the phenotype of the cell or the phenotype of the organism may be affected. Right, so this is how phenotype is affected by a change in the DNA of the gene. Okay. Right, so let's look at addition and deletion first. Right, this is when one or more base pairs are either added or deleted, right, from the gene sequence. Right, so this is the gene sequence, right, and I've highlighted the template strand as well. Right, so after transcription, this should be the mRNA sequence, right, and the mRNA is of course read in codon, starting with the start codon AUG all the way to the stop codon UAA in this case, right, to give rise to a polypeptide chain, right? So imagine there's an addition of 3 base pairs now, right? Addition, mutation, and addition of 3 base pairs, okay, to the gene, right? So you can see the added base pairs in red, right? Addition of 3 base pairs together, right? So what happens is after transcription, there will be an additional codon, right? So that would also be translated, of course, right? The ribosome won't skip the codon, right? So there would be an addition of an extra amino acid, right? But the reading frame is the same, right? It does not affect the rest of the codons, right? So you have the same amino acid sequence with the addition of 1 more amino acid, right? So that is the result of an addition of 3 base pairs, right? If you think about deletion, then a deletion of 3 base pairs would result in a deletion of a codon, and therefore 1 less amino acid, right? Unless you delete the start codon. Right, but if we have the addition of not 3, but 1 base pairs in this case, right, as shown in red, so what happens is after transcription, right, there will be 1 additional nucleotide in the mRNA sequence, and then the reading frame would change, right? Again, the ribosome won't skip 1 nucleotide, right? So it will read after AUG, AUU instead of the original UUG, right? And then instead of the original GCA, it becomes GGC, and so on. So the reading frame is shifted by 1 nucleotide. Okay? So that it will cause the frame shift mutation, right? So the amino acid sequence at the amino acid sequence after the site of mutation would all change. Same for deletion, all right? If nucleotides are deleted, right, either 1 or 2 even, right, then the nucleotide sequence of the mRNA would be shorter, right? And then the reading frame would change also. You can imagine that yourself. Okay, so the basis is that if addition or deletions are not in multiples of 3, right, then that would cause frame shift. Okay, if addition or deletion is in multiples of 3, then you either add 1, 2, or 3 codons, or you delete 1, 2, or 3 codons, right? As long as it is addition of, say, 3 nucleotides, 6 nucleotides, 9 nucleotides. But if it's not in the multiple of 3, then that would change the reading frame because the codons are read in 3s, right? And that would be frame shift if the reading frame is changed due to the addition or deletion of not multiples of 3 base pairs. Okay? So next we look at substitution, which is when a nucleotide or base pair is replaced. So there's not addition or deletion, it's replacement, substitution. Okay? So same example as just now, right? You have the gene sequence, the transcribed mRNA sequence, and the translated amino acid sequence here, matching to the codon. Yeah. So if there is a substitution here, right? For example, and it's this change to this nucleotide instead, or I can see IGC change to TA. What happened to the mRNA is the codon is changed from GCG to GCU. All right. If you look at the codon table under the genetic code, GCU would code for the same amino acid. So there's no change to the amino acid sequence, right? So this is known as a silent mutation as a result of the substitution mutation. Okay, no change to the amino acid sequence, so therefore no change to the protein itself, right? And that's due to the degeneracy of the genetic code, right? The two different codons, but coding for the same amino acid. Right? Another scenario, right now we substitute this base pair, right, from CG change to AT. So after transcription, what happened, right, you have AAC instead of CAC, and AAC would code for a different amino acid, right? So instead of histidine, it is now asparagine. Okay, so the changed codon due to the substitution mutation, right, codes for a different amino acid, and this is known as a missense mutation. So the polypeptide sequence is changed by one amino acid, right? So it may or may not change the shape and function of the protein depending on how important this changed amino acid is, right? So it is if the histidine at first, right, the original amino acid is found in the active site, for example, and now you change the amino acid, right? Then the enzyme may not be functional, right? Because amino acids at the active site, of course, play important function, right? Either to bind to the substrate, if not to catalyze reaction, or if this amino acid is important in the folding of the polypeptide, it may be forming a interaction between R groups that's important to maintain the shape of protein. Then changing the amino acid may affect folding. Or if the original amino acid actually carries, say, a hydrophobic R group, right? So it should be found in the interior of the protein, right, after folding. Now the new amino acid may, if it contains a hydrophilic R group, right, then the folding would also be affected, right? Now the hydrophilic R group cannot be found in the interior, right? So the folding of the polypeptide will also be changed. Right, so there are scenarios when a change in one amino acid may change the shape and function of the protein, but of course there are scenarios where there's no change, right? So the protein still function normally even though there's a change in one amino acid, right? So this may or may not affect protein function. Okay, and this is a missense mutation, right? And last scenario, right? You have a change of nucleotide here, right? TA becomes AT. Okay, so the result is the codon is changed from UUG to UAG, and UAG is a stop codon, right? So this is a nonsense mutation when the codon is changed to a stop codon that causes the termination of translation to be earlier, right? So premature termination, right? And the polypeptide form would be shortened, so we call this truncated, shortened, right? So in my example, it's not even a polypeptide. My example is only one amino acid, right? But of course, in real examples, it can still may have a shorter polypeptide, but that would of course significantly change the amino acid sequence because now the polypeptide is shorter, right? And significant change to the primary structure, and therefore significant change to the shape and function of the protein. Normally non-functional. Normally the protein would be non-functional. All right. So these are the different examples of the mutations, right, in the gene, right? Whether it is deletion, addition, causing frame shift, may or may not cause frame shift, or the substitution that would result in either silent, missense, or nonsense mutation that may or may not affect protein function. Okay. Now a question, right? So we talk about mutations in the gene, right? But we know that for eukaryotes, the genes contains introns, which are removed, right, in post-transcriptional modification from the mature mRNA. What would happen if the introns are mutated? Or what will happen if gene mutations occurs in the introns? All right, so pause the video, think about it. Once you have some answer, you can resume the video. All right, so mutations, if it occurs in the introns, not at the splice site, then it does not really affect the mature mRNA sequence, right? Because whatever mutation that occurs would still be transcribed into the pre-mRNA, but would be removed after the introns are removed, right, during splicing. So no change to the mRNA sequence, the mature mRNA sequence, and therefore no change to the protein itself. But if mutations occur at the splice site, right, remember the splice sites are important during the splicing process because they are recognized by the snRNPs. Right, so if the mutation occurs at the splice site such that snRNPs cannot recognize the intron anymore, right, then that will affect splicing. And that would, of course, change the mature mRNA sequence. Then that would change the protein. Right, there will be a change in the polypeptide sequence. Okay? Right, so imagine if an intron is not removed, then your mature mRNA is longer. Then the protein may be longer, and that will significantly affect the protein function. Or if the end of the intron is not recognized, right, and then the splicing complex, the spliceosome, actually remove not just the intron, but the exon after it as well, because it doesn't know that the intron has ended. So the intron, the exon behind the intron, would be removed together. Right, then that would shorten the mature mRNA sequence. There's a removal of one exon, right? And that would, of course, also change the protein length, protein structure, and function. Okay, so introns, even though they are removed, right, mutations to the introns may still affect the protein if it occurs in the splice site. All right, so that would be the end of this video.